Okay, so we have the last portion of this lecture, which will be chromatography. Today, I'm going to talk briefly the fundamentals of chromatography. And on Wednesday, we will have a brief introduction to HPLC so that next week you can start, your lab instructors can help you to start the HPLC lab. And on the coming Monday, we will have our last lecture about gas chromatography. Now, so for the Wednesday will be our last, the coming Wednesday, no, the next Wednesday will be our, sorry, next Thursday will be our last lecture. So I expect that I will give you a review of the whole semester and for the final exam. You probably can check your final date on your GET, but I believe it's December the 17th. December the 17th, starting from 9:30 to 11:30, I think. That's the scheduled time, okay? All right, so we're now we're studying to talk about the introduction to analytical separations. I do want to remind you that even though the title here is analytical separations, and you will notice that in the lecture, I will just be talking about chromatography. However, chromatography is not the only analytical separation techniques. There are also other analytical separation techniques, for example, extraction, electrophoresis, separation. So we're just not covering that part, but keep in mind that there are different types of analytical separations, okay? Now, for today's lecture, we will just focus on chromatography. What is chromatography? So if you look at the name of this, if you just look at the word here, chromatography, it means color writing, right? The name was invented by a Russian botanist who actually developed this phenomenon by using a calcium carbonate solid phase to separate several plant pigments. Okay, this is how the setup works. So he filled the tube with those calcium carbonate solids, and then he poured a solution containing those plant extracts. And when the solution goes through the column here, the different pigment gets separated because different pigment will absorb to the solid phase at different strengths. Some of them will absorb strongly, some of them will absorb weakly. So those that absorb weakly will flow out first, which is why you can see that originally it's a mixture of all the pigment, but during the process you can see that different pigment can be separated. Okay, so this was this phenomenon, and this chromatography term was developed in around 1900 by this botanist. And then later on, around 1940s and 1950s, another two scientists actually further developed this technique by developing the detailed fundamentals and methodology for partition chromatography, which makes this technique, which is the foundation to make this technique widely applied in later research and industrial usage. So because of that reason, these two scientists received the Nobel Prize in 1952 for this development of partition chromatography, okay? And when we are talking about chromatography, there are two very important terms here. They're called stationary phase and the mobile phase. So taking this one as an example here, we know that the stationary phase would be the calcium carbonate filling inside the tube, right? Because those are staying in the column, in the tube, they're not moving out. Versus the solvent used for containing those plant extract are called mobile phase because they're moving, they're mobile. Okay? And you will see that depend on what type of stationary phase, what type of mobile phase you're using in the equipment or the design, there are different types of chromatography technique. Okay, so first of all, if we are trying to classify the different chromatography techniques based on the mobile phase, we can have gas chromatography or GC or liquid chromatography, LC. And you're probably familiar with the term HPLC, which is high performance liquid chromatography. And a relatively new one, supercritical fluid chromatography, is that if you look at the phase diagram, we have the gas, we have the liquid, and supercritical fluid is kind of in this region. There's no clear boundary between the physical properties between gas and liquid in this region. So it's called supercritical fluid. The reason that people find this could be very important application here is that when you use supercritical fluid, a tiny tuning of the condition can change the property very largely. Okay? So we're not going to talk about the details for this. For this lecture, as I said, on Thursday, we will talk about HPLC, and on next Tuesday, we will talk about GC gas chromatography and their applications briefly. Okay? And this class, because it's an introductory level course to analytical chemistry, so we're not going to talk a lot of the details about the instrumentation. If you're interested in those techniques and learn more, you could take the 4520, which is focusing on chromatography and mass spec